Hello, my name is Luis Concha. I am from the Institute of Neurobiology at the National Autonomous University of Mexico. And today, I'd like to introduce you to MRI, magnetic resonance imaging. MRI is probably one of the most popular methods around in medical imaging. It is certainly very useful, and it's because of how it's made, because of how the images are acquired, it needs the combination of many different aspects ranging from physics, chemistry, math, and technology, and computer science as well. So because of that, there have been many Nobel Prizes awarded to people involved in the field of MRI, starting all the way from work that was done in 1944 in the description of the MRI phenomenon, all the way to 2003, when Lauterbach and Mansfield obtained the Nobel Prize for the way we do imaging nowadays in 2003 because of work they had developed in 1975. So as you can see, this is a very exciting field. You can get into it from many different angles, and you can extract from it whatever you like. And there's a place for everybody in the field of MRI. So the MRI phenomenon, which again was described in the 40s, is a phenomenon that occurs in nature in atoms that have an odd number of protons. That is, protons intrinsically have a spin, and because of that, because they are charged particles, they have an associated magnetic field associated with it that we're going to call mu, and it's shown here as this yellow bar that is rotating. So this yellow bar is a magnetic field that when it's in the presence of a very large magnetic field that we're going to call B0, and it's highlighted as this green large arrow, this mu or little magnet associated with the proton starts to move, so it aligns with the main magnetic field, B0. But not only that, it starts to precess around it. And this term precession is exactly what you're seeing in the animation. It is essentially like wobbling around the main magnetic field, like a gyroscope would wobble around the axis of the gravity when it starts to topple down. And the frequency of this precession, that we're going to be calling omega, is relative to the magnetic field B0, and a constant that depends on each atom that has the property of MR precession, that we're going to be calling gyromagnetic ratio, or gamma. So as you can see, the frequency of precession depends only on these two factors, and different atoms precess at different frequencies. That is, they all have different gammas. And really, for this talk and for 99% of applications in MRI, the gamma that we care about is that of hydrogen. And for hydrogen, gamma is 42.5 megahertz per Tesla. So that means that for every Tesla that you have, this little bar magnet mu is moving around B0 42.5 million times per second. And as you know, MRI magnets have different field strengths that range from 0.5 to 3, 7, or even 11 Tesla. So a 3 Tesla scanner, as we can see down here, is a very, very powerful magnet when we compare that to the magnetic field of our planet, which is roughly between 30 and 50 micro Teslas. So that is a huge difference. And we know that even such a small field can provide enough energy or enough traction, let's say, to pull the needle in a compass and make it point towards the north. So a 3 Tesla magnet is incredibly powerful. If you get close to it with your keys, for example, it will yank them off your hand, and they can become projectiles. So it is very crucial to be absolutely careful with whatever you're carrying into the MRI room. The MR magnet never sleeps. It's never turned off. It's always on, and it will pull whatever magnet you bring into the room, into the magnet. So every time a patient comes in, every time an MR tech walks into the room, there's a screening that needs to be done for every person, so that you make sure that there is no metal either inside the body, say a prosthetic hip replacement or a pacemaker, and there's also no metal outside, like no coins in your pockets, you don't have a pair of scissors, a tool, etc. So B0 is the main magnetic field, and if we're talking about MRI, nowadays in 2024, we're generally talking about human scanners with a 1.5 or 3 Tesla field strength. There are higher, but these are two of the most commonly found throughout the world, 1.5 and 3 Tesla. So now let's talk about hydrogen, because that's essentially where we want to get our signal from. And if we put as an example a cube of 1 millimeter per side in the cortex of the brain in a human, there are roughly 310 times 10 to the 19 water molecules, and each one of those will have 2 hydrogens that will be able to provide us with whatever signal we're going to obtain and use in a few minutes to get a signal. And again, in that same little cube, we have roughly 250,000 neurons that we're going to be studying and are going to be averaged in terms of signal in that little 1 millimeter per side cube. So this is the water molecules and the hydrogens in them that are going to be providing most of the signal that we care about in MRI. So how do we get this signal? So we need to discuss the MRI phenomenon, or the MR phenomenon, I should say. So in my representation, we have these protons as these orange spheres, and they have their little bar magnets that we called mu a while ago associated with them. But because each one of them is so weak, they're all pointing to whatever orientation they want, and they're all interacting with each other, and they're really not aligned in a particular orientation. But when you put them inside a main magnetic field, now again, it's called B0, then suddenly they all align to this main magnetic field B0. So they're neatly organized, they're all pointing in the same direction, and not exemplified here because this is not an animation, but they are precessing around B0 as we saw before. So if we know the frequency of precession, omega 0, that we noticed before, then we know exactly the frequency that we have to emit through a radio frequency that, if we recall, we were in the range of the megahertz, so that corresponds to frequency modulation, FM radio. So this is not ionizing radiation. This is extremely non-harmful. Nothing will happen. There's no risk of cancer or mutations or anything like that. But what it does is that if you provide this radio frequency at exactly the frequency of precession, which we derived from the formula we saw before, then we're going to attract all these little bar magnets and make them point towards the radio frequency emitter, the antenna, the coil. So now that they are pointing to us, we can turn off this radio frequency, and naturally, these bar magnets will return to their original position, that is pointing towards the main magnetic field, B0, and in doing so, they will release back this extra energy that we had provided in the form of radio frequency, and they will emit this signal back again in the range of FM, and we can collect that with our antenna, with our coil, and get all this signal back. Now, we provided some radio frequency, and we're getting radio frequency back. So the interesting bit is whatever happened to this signal while we threw it into the tissue and then it returned slightly modified, and these modifications are we're going to be talking about the nature of the tissue. That's what we care about. So when we get this signal, is going to be a very complex signal that comes from millions and millions and millions of different water molecules and millions of neurons. And it's going to be very complex if we plot it over time. We can see that it decreases over time. That's interesting. But if we look at the frequency content of this, it's extremely complicated to wrap your head around. But we have a very fantastic tool that was developed a very long time ago by Joseph Fourier. This is the Fourier transform. And the Fourier transform says that we can get any signal that happens over time, and we can decompose it into very simple periodic signals that, when added all up, will result in the original signal. So this is like a chord in the piano, right? So you can play C, G, and E at the same time. It will sound like a chord, but the Fourier transform says that this is just 3 keys that are pressed in unison. And not only that, the Fourier transform can tell us that we actually pressed the key of C a little bit harder than we pressed the other 2 keys. So that's what the Fourier transform says. And because the frequency of precession depends on the magnetic field that we are experiencing, any modulation of the magnetic field will result in different frequencies that we can extract using the Fourier transform from this original signal that we obtained. All right, so we got some signal, but we don't have an image. And this talk is about MRI, I as in imaging, right? So the crucial aspect is to remember that the gyromagnetic ratio tells us the frequency of precession of these spins of mu around B0, and that relies, if we make this gamma a constant, which is the gyromagnetic ratio, it's a constant, then really the only thing influencing the frequency of precession is the main magnetic field strength, B0. And again, we are looking at 42.5 megahertz per Tesla. And if we have 3 different samples in a scanner and we want to know where they are, if we put the scanner at, say, 3 Teslas, but we slightly modify the magnetic field strength from the left hand of the image to the right hand of the image using what we call a magnetic field gradient, that is a linear and user-dependent shift of B0, we can make this right-hand side have a little bit higher magnetic field than on the left. And because the magnetic field strength is a little bit higher on the right-hand side, we are processing at a slightly higher frequency on the right-hand side than on the left-hand side. And again, here we have a little bit less than 3 Tesla. We have 2.99 Tesla. And by plugging in the equation, we have 127.075 megahertz. So whatever sample we have here that has hydrogen and water is processing at a slightly slower frequency, slightly lower frequency than what it's processing on the right side. And we're going to get the signal back with our antenna. So we're going to excite our tissue with the frequency of precession, and we receive this different frequencies, or the signal that is actually composed of different frequencies, because there's something on the right processing at higher frequency than something on the left processing at lower frequency. And the Fourier transform will tell us that indeed that is actually what is happening. We have something that is processing at high frequency and actually has a high amplitude of the signal because there's more water in that sample than whatever is processing on the left-hand side, and the middle processing at different frequencies, and the amplitude of the signal tells us how much signal there is at that particular position. So what we just did is to spatially encode tissue or components of tissue or our sample. Now we have an idea of where things are, at least in one dimension, and it's all based on the frequency of perception. Now, that is a very clever idea, and these sorts of ideas is what gets you a Nobel Prize, because this is a basic concept of MR imaging. We just looked at one dimension, and we need to encode three because our bodies are composed of, left, right, inferior to posterior, superior to inferior. But the basic idea for encoding spatial position in any of these three dimensions is essentially this one. It's just little neat tricks that are done to spatially encode the other two dimensions, but it is the frequency of precession that we can modulate with magnetic field gradients, here in orange. That is what is causing the different frequencies of precession, and in turn informing us of spatial locations. Now, we need to do more things to get an image. So we just looked at one dimension, and if we want to encode the three dimensions, then we're going to be doing this little trick with neat tricks added to it over time. So this is pretty much like a musical record or a musical notation in which different notes happen at different moments in time, and we call this a pulse sequence. So in a pulse sequence, you have your different ingredients on how you're going to make an image. For example, our first channel is the radio frequency, and it tells us how much radio frequency we're going to emit at one point in time, and then another point in time, and then how we're going to play out our magnetic field gradients, if we're going to be shifting the magnetic field from left to right or anterior to posterior or in some diagonal. When are we going to do this, and when we are going to get our signal down here at the bottom? All of these are parameters that we can change. We can change their timing, we can change their shapes, and the way we add up all these ingredients make a different musical record, and these different pulse sequences allow us to obtain different forms of imaging and different forms of contrast that we can ask different things about tissue. Now, if we just get an image and it's all white, that's pretty boring, right? And if it's all black, it's even worse. So what we need is some way to obtain contrast in the image so that we have some pixels that are bright white and some pixels that are gray and some pixels that are dark. So we are going to rely on phenomena that are intrinsic to the tissue that we call relaxation. And relaxation we saw when we turned off the radio frequency and the spins looked back at the B0 at the main magnetic field. And the rate in which this happens is called relaxation, and different tissues will have different rates of relaxation. Now, there are two different forms of relaxation, but they occur at the same time. One is longitudinal relaxation that we call T1, and that occurs in the range of seconds. And the other is transverse relaxation, which occurs in the 10 or 100 millisecond rate, and we call that T2. Transverse relaxation, T1, is quite simple to understand. That is because when we made all the little mews looked at the antenna when we were emitting the radio frequency coil, the radio frequency at the frequency of precession, and then we turned it off, they will try to look back at B0, which is aligned with the Z axis on our grid. And in doing so, it will display this very interesting trajectory that looks like a spiral going upwards, pretty much like an ice cream cone. And if we plot the projection of this magnetization on the Z-axis, we will see that if it was completely perpendicular to it, it will slowly, it will gradually grow all the way back until it's fully relaxed. And in doing so, it will display this exponential ascending curve. And it is this exponent that dictates the shape of this curve that we call T1. And different tissues will relax faster and some tissues will relax slower. And that all depends on the ability of the tissue to dissipate the excess of energy that we deposited in the form of RF, radio frequency. And as you can imagine, fatty tissues like white matter, like fat, will dissipate this excess energy much faster than water can. For example, the cerebrospinal fluid in the brain, and that has a very slow relaxation rate. Fat, again, will display a very quick relaxation, and water will have a slow relaxation. It is these differences in relaxations that will provide the contrast, or one of the contrast mechanisms in our images. The other contrast mechanism is T2, or transverse relaxation. Here we need to think of ensembles of spins that are trying to process all at the same time. And as you can see, I have three different little bar magnets here that are processing right after we turned off the radio frequency coil. So these are aligned on the XY plane. They should all be processing at exactly the same frequency, but because the tissue has some properties that make whatever spin is on the right, a couple micrometers on the right, then another spin on the left, they are processing at slightly different frequencies, and hence they tend to dephase. They are no longer spinning at exactly the same phase. One of them, the orange one, is starting to get ahead of the yellow and the green spin, and the green is lagging behind. And as you can see, they're starting to fan out in terms of how they are organized within each other. And these dephasing, if we think of it as a vector sum, the transverse magnetization that we have on the XY plane is going to tend to decrease over time, again in an exponential fashion. And it is this exponent that is dictating T2 contrast. And the result is the loss of signal over time. And that is very interesting because, again, different tissues will have different rates of decay. So here we have three different tissues in the brain that are very important, and we're displaying their different relaxation rates in terms of T1, T2, and DP is called proton density. Sorry, this is in Spanish. It should be PD, proton density. And that means essentially the level of hydration of tissue, with CSF being essentially water, and white matter being a little bit fattier, it has less water. And we can see that different tissues will have these different curves, with white matter having a very fast longitudinal relaxation, and also a very fast signal decay because of T2, and CSF being exactly the opposite. It has a very slow longitudinal relaxation, and a very slow transverse relaxation, and a very slow loss of signal on the XY. So it is these differences that allow us to obtain contrast in the images. All of that we've seen, how we're going to emit the radio frequency, how it's going to change because of the D0 differences, how long it's going to take to decay, how long it's going to take to relax completely. All that can be neatly described through the block equations. So that's a lot of math. It's very interesting, but it's a very compact way to explain the evolution of signal after whatever you want to do with your imaging, whatever pulse sequence you want to do with it, can all be neatly predicted through block equations. And this is the math and the physics that are behind MRI, and it's all a very interesting avenue of study. Now, going back to simple images, we can see that if we play with our pulse sequences, and if we play with the parameters of pulse sequence, we can obtain incredibly different contrasts in images. So here I am showing you three different images that are acquired with a particular pulse sequence. that by changing just one parameter that's called echo time, and we use the acronym TE, TE in English as well, as in Spanish. Just by changing this echo time, we can obtain an image that has very different characteristics, ranging from one that is having all tissues having very bright signal in the CSF and the ventricles of the brain, and also in the white matter that is something slightly darker. But if we move this parameter from 18 to 100 milliseconds, we then get to having an image that has very bright CSF and very dark white matter. And that is happening exactly because of one of these plots that we saw here, showing that CSF takes a very long time to decay in terms of signal in the transverse plane, whereas white matter has decreased its signal very quickly, and we were at 100 milliseconds, so roughly here, and that's exactly what we can see here. CSF still showing us a lot of signal, and white matter showing very dark signal. So that's the very basic mechanisms, or one of the basic mechanisms to obtain signal and one type of image. But the beauty of MRI is its versatility. We can obtain images of the brain, we can obtain images of the knee, of the entire brain, of the entire body. We can look at musculoskeletal images to look for tendons, bones, ligaments, muscles, etc. We can also look at the brain functionally and try to see which areas of the brain are more active in different activities, and humming, singing, or speaking, and try to compare the different parts of the brain that are more or less active in one of these other activities. We can look at the connectivity of the brain via diffusion imaging and tractography. We can look at how one part of the brain connects to another part of the brain and try to understand what the flow of information is in the brain. We can look at the perfusion of tissue through the evolution of signal with a contrast mechanism that has been injected. And by studying these curves in every pixel of the brain, we can understand how much blood and how fast it's flowing into and out of the tissue. And we can even obtain dynamic images, as we can see here in the heart, that provide a tremendous amount of detail in the anatomy and function of the entire body. So MRI is probably one of the most versatile imaging techniques there are. It is non-invasive. That means you can have as many MR scans as you want, and there's really no problem unless there's any metal in or outside of the body. It's pretty much harmless. As we can see, it's extremely versatile. It has very high resolution. It can give us a lot of detailed images. We can make it quantitative by studying parameters and comparing from one point to the other. The downside is that MRIs are expensive. Not all places can afford an MRI. They have gotten cheaper over the years, but they are still a very heavy investment for any hospital, for any institution. Imaging is relatively slow. A typical MRI scan will last between 15 to 25, 30 minutes in a hospital, and in research scenarios, it can range up to 1 hour maybe. But regardless of these two, MRI, I think is super cool, and it is an area of study that any person can get into and find something that they will like, either if they are physics-oriented, math-oriented, or biologically oriented. MRI is incredibly cool. So that's it for this introduction. I hope you learned, and if you have any questions, please send me an email. Thank you very much, and goodbye.